All right, so today we are going to finish talking about some of the organelles. Like I said yesterday, and this year we're going to talk about more detail about each of them, rather than just saying sort of what their function is. We'll talk more about their structure, what they do, how they interact with other organelles. So we'll just get into a little bit more detail than we did back in 7th grade. So as we just talked about in question today, the nucleus is basically a container that holds the DNA, the chromosomes, the genetic material. So the nucleus is surrounded by its own membrane. Sometimes it's called the nuclear envelope. And inside of that nuclear membrane are the chromosomes, the genetic material, the DNA. And it takes different forms. When a cell is sort of in its in-between phase and it's just sort of doing its activity or it's growing, you can actually see individual chromosomes inside of the nucleus. The DNA has been sort of unwound. It's not condensed into chromosomes. When it's in that form, we call it chromatin. When it winds up and forms like specific strands that we can actually see, then we call those chromosomes. But it's the same material, it's just in a different form. The nuclear membrane, the nuclear envelope, actually has small holes in it, small pores. I'll show you what they look like in a minute. You can see one here. And so we know that the DNA itself can't leave the nucleus. Okay? It can't get through that nuclear pore, but smaller substances can make it through. Okay? So RNA, ribosomes, they can get through those pores that are in the nuclear envelope and move out of the nucleus to the cytoplasm. Also inside of the nucleus is an area called the nucleolus. It stains darker. And so when you look at a cell in the microscope, if you can see the nucleus, a darker area within the nucleus is called the nucleolus. And it's a mixture of DNA, RNA, and proteins. What happens in that area of the nucleus is that's where the ribosomes are made. The ribosomes are produced in that nucleolus, and then they can leave the nucleus through the pores in the nuclear membrane out into the cytoplasm or to the ER, so they can start fulfilling their function of protein synthesis. When a cell divides, and we'll talk more about cell division at the end of this unit, the nuclear membrane and the nucleolus both sort of disappear and are no longer visible when the cell's in the process of dividing and reproducing. So if we look at this large purple thing in the illustration, that's the nucleus as a whole. The nucleolus being the darker area in there, and you can see the DNA in this form is chromatin, just all sort of spread out and diffuse throughout the nucleus. It has the membrane around the outside. It's a double layer membrane, kind of like the cell membrane. And embedded in that are these little pores, these little openings. So these are illustration, but if you look at an actual electron microscope images of the nucleus as a whole, you look at the surface with greater magnification, these pores, these little circles, you can see them in there. Those are the pores where things can move in and out of the nucleus. Those ribosomes that are produced in the nucleolus, they're made of RNA, ribosomal RNA. And so that's shown here in sort of blue is the ribosome. They have two what are called subunits that kind of bind together to form a ribosome. And when you have these ribosomes attached to a strand of messenger RNA and transfer RNAs bringing in amino acids, this is how proteins get produced, by these three types of RNA working together. Ribosomal RNA, the transfer RNA, and the messenger RNA work together to build up proteins one amino acid at a time until they've been synthesized. So these ribosomes, some of them are just floating in the cytoplasm. These ribosomes are not attached to anything, they're just in the cytoplasm. Other ribosomes are actually attached to the ER. They're embedded in the surface of the ER, which we'll talk about in a minute. And those different ribosomes, depending on their location, they have different functions. The ribosomes that are attached to the ER, they typically are synthesizing proteins that are going to be staying inside of that membrane or are going to be leaving the cell. The ribosomes that are just floating around in the cytoplasm, they generally make proteins that are going to be used in the cytoplasm, within the cell, which makes sense. So that ER, these long tubes, is another organelle. It's basically a series of membranes within the cell. Here you can see this is the ER, this sort of tube here. You can see that has some of these ribosomes attached to it. And having this series of membranes within the cell provides a large amount of surface area where chemical reactions can take place. There's two types of ER. They're called the smooth ER and the rough ER. The smooth ER is ER that doesn't have ribosomes attached to it. And so therefore, the smooth ER doesn't help in the production of proteins because it doesn't have those ribosomes. Instead, in the smooth ER, things like lipids and steroids, phospholipids, they're produced in the smooth ER. Also, this is where toxins can be broken down, detoxified, things like drugs and poisons are broken down by the smooth ER. And so tissue like your liver has an abundance of this because that's one of its functions, to detoxify certain substances. The rough ER is where the ribosomes are attached. It's called rough because it looks like it's rough with all these ribosomes embedded in its surface. And so the rough ER's job is to produce the proteins along with the ribosomes. And oftentimes these proteins produced there might be getting shipped out of the cell. We'll talk about how that happens in a minute. For example, insulin. So insulin, the hormone, is produced within cells. Cells need to produce quite a bit of it, and it needs to be released into the blood at the right time. So insulin is produced by a ribosome attached to the ER. The insulin can be stored in the ER until it's time to be released, and then it can do that with the help of the Golgi apparatus. So here you see often the ER is typically near the nucleus. Here you see the outer layer of the nuclear membrane. And then sort of attached is the layers of the membrane for the ER. Smooth ER and rough ER look different. So here we have the rough ER. You can see some ribosomes attached. Here's what it actually looks like in the electron microscope. This is the rough ER. And then the smooth ER looks a little bit different. It doesn't have the ribosomes. It also is a different sort of structure. It has these branching shapes that you can see in the smooth ER over here. And so materials that are made in the ER can move through this series of membranes from one to the other. And eventually, when it reaches the outer edges of the ER, can be packaged up into little sort of vesicles, which can take it to the Golgi apparatus. Golgi apparatus is where these materials are going to be prepared and then secreted from the cell. So when these proteins that are produced in the rough ER get to the Golgi apparatus might modify those proteins in some way, might store them up until they're going to be released. So it looks like a flat end stack of discs, and it has sort of two sides. One side faces the ER. That's how the cis face. So materials from the ER come to the Golgi apparatus from this side. And then there's these series of tubes. The other phase, the phase is sort of the outside of the cell, is called the trans phase. And so as materials come through, they get packaged up in these little bubbles called vesicles. And these vesicles now are headed out of the cell. You can't see the outer edge of the cell, but it'll be like down here somewhere. So here again, you see material from the rough ER coming in to the Golgi apparatus, moving through the Golgi apparatus until on the trans phase it gets packaged into these vesicles where it can move out to the cell. So for example, if these were cells in the pancreas, there would be a lot of this Golgi apparatus, and these little blue spheres might represent insulin as a hormone. And yeah, these are the pancakes with the drops of delicious golden syrup. That's how you describe it in some degree. That's what it looks like. And these things are getting ready to leave the cell. Golgi body, Golgi apparatus, yes, same thing. Just two different terms for the same thing. So you remember that, that's good. All right, lysosomes. What did that root lyse mean? Like hydrolysis, lysis.
Oh, it's lysosome. Breaking something apart.
Yeah, break something apart. The lysosome has hydrolytic enzymes. Enzymes that are going to break things down. Digestive enzymes. They're just small little vacuoles that hold these enzymes that are responsible for breaking down material. They're important for several reasons. In cells that have to destroy pathogens, for example, like white blood cells, these lysosomes can allow that to happen. They have enzymes that can break down a foreign cell. Or if large molecules need to be broken down, these lysosomes can help that to happen. If cell organelles are no longer useful and need to be sort of recycled, lysosomes can break them down and then the cell can reuse those materials. So they're not very recognizable, they just look like vacuoles, small little vacuoles. So here's an example of how a lysosome might help an organism. So here you have some food particles that this cell has ingested. What kind of ingestion would you call that? Yes, penocytosis. It's a pinching in of the cell membrane. What was the one when it engulfs? Phagocytosis. But anyway, here a food particle has been ingested by penocytosis. Now it's inside of the cell. It's in a vacuole. Here are lysosomes. These little pink things inside of them are the enzymes. And so a lysosome could then merge with this food vacuole. The enzymes then can break down that large food molecule into its components so the cell can use them. That's how a lysosome works. Vacuoles are just storage areas. They're just a small membrane within the cell storing something. So in unicellular organisms, there are some specialized vacuoles, like a food vacuole, that allows for the breakdown of food molecules that have been ingested. A contractile vacuole, you may remember from 7th grade, protists have it, which helps, it pumps water out from freshwater protists because they constantly have water coming in through osmosis. This contractile vacuole fills up with water and then pumps it out. Help maintain water. Also, plant cells have a large central vacuole. Here we see a transmission electron microscope image of a plant cell. And this big area you see in the middle looks hollow. That's actually its vacuole, its central vacuole. And it's filled up with water. And when it's completely filled with water, it's exerting pressure on the cell wall around the outside. And that helps the plant cell maintain its rigid shape. What happens if it's not filled with water? It shrinks, and what do you think happens to the rigidity of the cell? Yeah, it sort of starts to become less rigid. Okay? What do we call that? If a plant doesn't have enough water and it can't hold its shape? No? Well, there's another word we use for it. If you have plants and you don't water them, they will wilt. So they wilt. It means they can't stand upright because they aren't able to have that rigid shape because this vacuole has lost water. Like if you have a beach ball, if you let half of the air out, it's going to be sort of floppy, it won't hold its shape. Versus if you blow it out completely, the pressure pushing out against the walls will help it maintain its shape. Same sort of thing with these plant cells. The vacuole is filled with water, pushes against the cell wall, and they can hold their shape. Yeah. Mitochondria. The organelle that's responsible for releasing ATP for the cell. It is the site of cellular respiration. It takes place in the mitochondria. They look like beans with a bunch of folds inside of them. They have a complex shape. They have a double membrane. So the folds on the inside are called cristae. They increase the surface area of the mitochondria because there's a complex series of chemical reactions that take place, which is what cellular respiration is. As we talked about in evolution, mitochondria have their own DNA. They have their own ribosomes. They can replicate themselves. That was evidence for the endosymbiotic theory of how eukaryotic cells evolve. That maybe in the far distant evolutionary past, these were individual cells, prokaryotic cells, that developed this symbiosis with other cells. And typically, cells have a few hundred of them. However, cells that are very active and use a lot of energy, like muscle cells, have more. Also, as cells are stressed with by exercise, for example, if you are swimming and you're training, okay, the more that you use certain muscles, they will adjust by creating more mitochondria in those cells. And so muscular endurance can be improved by repeated exercise. That your muscles will produce more mitochondria, allowing them to release more energy, allowing you to go longer and still maintain that energy in those muscle cells. So training, as an example, can encourage cells to produce more of these mitochondria. Okay? Yeah, the matrix is a liquid. It's the inside. Some parts of cellular respiration happen in the matrix. Some parts happen in the membrane. Chloroplast is similar to the mitochondria in that it also has its own DNA, its own ribosomes, and may have evolved from a photosynthetic prokaryotic cell. It's where photosynthesis takes place in plants and in protists. There are these little ovals inside. They have these stacks of discs. They're called thylakoids, those discs. And they have the pigment chlorophyll in them. Chlorophyll is a green pigment that can absorb light, and that is important in photosynthesis. The stacks of these discs, they look like poker chips or like macaroons or something. They, those stacks are called grana. And then the fluid that's surrounding them is the stroma. You can see them here in these compound microscope image of the plant cells, small circular organelles. If you look at them in sort of cross-section under the electron microscope, here are the stacks of discs in those. So they're responsible for photosynthesis. We'll talk a lot more about photosynthesis and respiration at the end of this. So that's the actual, that is the protein. So if you look at this, this is what's called a motor protein. It's a protein that allows for movement of things within the cell. And what it's walking on, that strand that it looks like a cable, that is a part of the cell's cytoskeleton. That's a microtubule. And the cytoskeleton is a network of fibers sort of stretched across the cell through the cytoplasm that helps support the cell. Especially, for example, animal cells. Because animal cells have that flexible cell membrane, you can imagine if they had nothing holding their shape together, they would just kind of flatten into like a puddle. But these fibers of the cytoskeleton are sort of stretched across, allowing them to maintain some sort of shape. They're also responsible for how cells move and how things within the cell move. So for example, if a vacuole is going, those vacuoles we saw, they're going from the Golgi apparatus out to the cell membrane. Well, they don't like swim there on their own. They're actually dragged there by things like these motor proteins. These motor proteins attach to the vesicle here. This might be one of those vesicles that contains insulin, for example. And the motor protein sort of attaches to the vesicle. The molecules on the bottom bind to this microtubule, one after the other, and it sort of walks this vesicle to where it needs to go.
How does it work?
Walk.
Pick up its life.
Yeah, it's sort of the release, the breaking and releasing of sort of bonds between this, the parts on its feet, if you call them that, and the microtubule. It's the repeated breaking and forming of those bonds. And it would require energy. It would require some ATP. Now, we don't need muscles. You know, muscles work by the same kind of process, by movement of these structures within the cell. Muscle contraction is also due to the action of the parts of the cytoskeleton. So there is a part of the cell called the centriole. They look like two rigatoni at right angles to each other. And these are important in cell division. They help to separate the chromosomes when a cell is reproducing. You can see them in the microscope. That's our microscope image. Here's one that's sort of vertical. Yeah. And they're at right angles to each other, and they're each made of 9 what we call microtubules. Microtubule is sort of what the name says. It's a tube. Okay?
Is that the thing?
Yeah, that's what the motor protein was walking on, microtubule. It's a thin tube that is hollow, and it's part of the cell's cytoskeleton. It also makes up cilia and flagellum, and it serves as a trap for those motor proteins. Another part of the cytoskeleton are microfilaments. These are thin, solid strands of molecules. Okay, they're made of a protein called actin. And these microfilaments, they are what's important in muscles contracting. Okay? Muscle cells have many of these microfilaments that can contract and relax based on these bonds that form between them. Some cells have flagella, which are contain microtubules, and a flagellum acts sort of as a propeller, pulling a cell around. Single-celled organisms sometimes use them for movement, but also certain types of cells within multicellular organisms might have flagella, like the sperm cell has a flagellum, allowing it to swim to reach the egg cell during reproduction. Cilia are very similar to a flagellum, but there's many of them typically. They're relatively small. They're also sometimes used for movement by protists. But they're also found in multicellular organisms. For example, in our trachea, we have cilia that help filter out contaminants from the air that we breathe. In the fallopian tubes, cilia line the inside of the fallopian tubes, and as they move, they sort of guide the egg cell down the fallopian tubes to the uterus. But these are also made of these microtubules, parts of the cytosol. Now we have the cell wall. The cell wall is a sort of outer, non-living, rigid layer that's beyond the cell membrane. It helps to give shape to certain types of cells. They have openings, so it's not like the cell membrane in that it's not really selectively permeable. It has passageways which things can move through it. And plant cells have a cell wall. So do other types of cells as well. For example, plants and algae, they have a cell wall made of cellulose. But there are other materials that can make up a cell wall. Fungi have a cell wall on their cells made of a material called chitin. Bacteria have cell walls made of materials like peptidoglycan. And so here in this microscope image, you can see this whitish layer on the outside. Those are the cell walls of these cells. That's the end of our cell organelle discussion. Technically, an organelle, often we will say membrane-bound organelles. In that definition, like a ribosome is not really an organelle. Cytoplasm is not really an organelle because it's not surrounded by its own membrane.